So my name's Sarah. I'm one of the nurses and patient care assistants at IVF-Life in Alicante. And this is María Enciso, who is specialist in reproductive genetics, human embryology, and assisted reproductive techniques. And she's also got a master's in clinical embryology and a PhD in cell biology. And she's the scientific director at iGLS, which stands for Integrated Genetic Lab Services. If anyone's interested, we can see it here on the first slide. And this is one of the labs that the IVF-Life group work with, and they also work amongst with other clinics as well. So let me just have a little look and see. So a couple more people are joining. So the webinar, obviously, Maria is going to go into more detail, but it's going to be all about PGT-A testing and the importance of the embryo quality in terms of the success in the treatment and the key role of genetics. So she'll go into a little bit more information shortly. I also just want to mention before we get started that we have a special collaboration with iGLS at the moment because May is considered Mother's Month. So we have a special promotion. You might have already seen that already on Instagram and things like that. So we're offering free PGT-A testing when you book your consultation before the 31st of May. So the first visit has to take place before the 30th of September. So yeah, any questions about that, we can answer them a little bit later. You should be able to see a little section that says questions and answers. So you can be asking your questions throughout the presentation, and then Maria will answer as many as she can at the end. And I'll have a little bit of input if applicable, but if not, then I'll hand over to Maria.
Well, good afternoon, everybody. Thank you very much, Sarah, for your kind presentation. Mm-hmm. For the next 20-25 minutes, we're going to go over a few slides that I've prepared to try to explain what is PGT-A, why is it worth doing this test, who can benefit from PGT-A testing, how do we perform the test in the lab, and I would like also to review some of the most recent studies that have reviewed the clinical value of PGT-A. And we will end up with some take-home messages of the main ideas that I would try to transmit you during this webinar. So I'm sure you all have heard about DNA. DNA is a molecule that is contained inside all the cells of our body. And this DNA is the molecule that contains the instructions for building an organism and to maintain this organism working. But what you may not know is that the DNA is organized in packages. It's organized like a book of instructions organized in chapters. And these chapters or these packages are have the name of chromosomes, which are this gray structure that you can see in the picture of the right. And the DNA in our species, the DNA in the human species, is organized into 23 pairs of chromosomes, which are the ones that are represented in this diagram. And what we can see is that each of the chromosomes inside each pair have the same structure but a different origin. One comes from our mother and the other one comes from our father. The first 22 pairs of chromosomes are called the autosomes, and they are named with numbers which are related to their size. Like that, chromosome number 1 is the largest in size, while chromosome 22 is the smallest in size. We have also the pair 23, which are the sex chromosomes, chromosome X and Y, which are responsible of establishing the sex, the biological sex in our species, XX for females and XY for males. All our cells have these 23 pairs of chromosomes except from the gametes. The eggs and the sperm have half the amount of chromosomes compared to the rest of the cells of our body. The gametes have just one copy of each chromosome. So sperm and eggs have 22 autosomes and 1 sex chromosome. And why these chromosomes are important? Well, during the process of this gamete formation and during the first divisions of embryo development, these chromosomes need to be duplicated and separated. And in the process of duplication and separation, errors can happen. Errors can happen so that gains and losses of chromosomes can be produced in the cells and in the embryos. And the presence of an abnormal number of chromosomes in a cell or an embryo is known as aneuploidy. And aneuploidies are a phenomenon that significantly impact the viability of a cell, an embryo, or an individual. Here you can see some examples of the chromosome complement of individuals with aneuploidies, with the gain or the loss of a chromosome. On the left-hand side, you will see the chromosome complement of an individual which has one chromosome loss. It has one chromosome less. If you look at the pair 23, the sex chromosome, you will see this individual has only one X chromosome. There is one Y or an extra X chromosome missing. And the fact that this individual has one chromosome less produces a disorder, a syndrome that is called Turner syndrome. On the right-hand side, you will see the chromosome complement of an individual which has a chromosome gain. In this case, it's affecting pair 18. So this individual, instead of having two chromosome 18, it has 3 copies of chromosome 18. And this causes a syndrome called Edwards syndrome. These are examples of aneuploidies that are compatible with life, that are transformed into individuals with a genetic disorder. But I would like to clarify that most aneuploidies, most chromosome gains or losses are not compatible with life, are incompatible with life. And the presence of these alterations in chromosome number in embryos are gonna produce miscarriages, are not gonna produce a pregnancy that arrives to term. What else do we know about aneuploidy? Well, we know that aneuploidy is very, very common in human embryos. It has been calculated that approximately 50% of all human preimplantation embryos are aneuploid. We also know that most aneuploidies have a maternal origin. They come from errors, for chromosome errors produced in the eggs. And aneuploidy, or the rate of aneuploidy, is very linked to maternal age. As you can see in the graph, aneuploidy increases dramatically with maternal age. You can see this, how this green line increases as the female age increases as well. It rises very sharply starting from 35 years of age. And what we can also see in this graph is the negative impact that aneuploidy has on reproductive outcomes. We can see that as aneuploidy increases in this green line, so does miscarriage rate in this yellow line, and inversely, the live birth rate drops. So the presence of aneuploidy is extremely detrimental to embryo viability and to reproductive outcomes. Well, now we know what is aneuploidy, how frequent aneuploidy is in human embryos, and how negative is the presence of aneuploidy for reproduction. But what can we do about it? Well, we can test the embryos for aneuploidy through an assay that is called PGT-A. PGT-A, this letter stands for Preimplantation Genetic Testing for Aneuploidy. And what is this? Well, this is a test that analyzes the DNA of embryos. We analyze the DNA of embryos before embryo transfer. Before embryo transfer, looking for what? Looking to detect an incorrect number of chromosomes in the embryos, so that only the embryos with a correct number of chromosomes can be selected for transfer, increasing the chances of pregnancy. Why is it worth doing PGT-A? Well, PGT-A is an advanced method for embryo selection. As you may know, in most centers worldwide, the system for selecting the best embryo for transfer is based on embryo morphology assessment. So looking at the morphology of the embryo, the best embryo of the embryo who is thought to have the most chances of developing into a baby is going to be chosen. Well, the embryo morphology cannot detect alterations in the number of chromosomes. And as we have seen, the chromosome content of an embryo is very important because only embryos with a correct number of chromosomes, this is with 23 pairs of chromosomes, are going to develop into healthy babies. Moreover, we know that aneuploidies are the main cause of implantation failure, miscarriage, and the production of a baby with chromosome disorders. So by selecting embryos with the correct number of chromosomes, we can significantly increase pregnancy rates. And who can benefit from this test? Well, PGT-A can improve the chances of achieving a pregnancy in all IVF patients. All IVF patients produce aneuploid embryos, so all can benefit from the selection or the deselections of these aneuploid embryos. However, PGT-A is especially suitable for couples that have experienced recurrent pregnancy loss, that have had previous IVF failure, women of advanced reproductive age, also couples that have already a family history of chromosome problems, or couples that are opting for a confident single embryo transfer. How is this test performed? Well, this test requires the removal from the embryo of between 5 and 10 cells at a stage where the embryo is at an embryonic stage, which is called blastocyst stage. This is 5 days after embryo development. And once these cells have been removed from the embryo, are introduced in a little tube and sent to the genetic lab. And in the genetic lab, we analyze the DNA of the biopsied cells and produce a report telling the clinician which embryos have a normal or an abnormal number of chromosomes, so that those embryos with a correct number of chromosomes can be chosen for transfer. I would also like to dedicate the last bit of the talk to show you some of the most recent studies that have shown evidence about the utility of PGT-A regarding several aspects, regarding pregnancy rates, regarding miscarriage rates, the time to pregnancy, or the embryo selection method for transfer. The first study I want to show you was this by Young and colleagues. This study was performed in good prognosis patients. This is young patients under 35 years old. And in this case, more than 100 patients were included in the study. These 100 patients were randomized into 2 groups. One group where the embryos for transfer were selected based on morphology. This is the no PGT-A group, and another group of patients whose embryos were selected by genetic testing, by PGT-A. What you can see is that in the PGT-A group, 425 embryos were tested, out of which 44.9% were found to be euploid and the rest aneuploid. This is with no options of transfer. So well, after this testing, what the authors find is that an ongoing pregnancy rate significantly higher of almost 70% is found in the PGT-A group compared to the no PGT-A group. So it seems that this selection method significantly increases the ongoing pregnancy rates per transfer. Another very interesting study is showing the clinical value of PGT-A in advanced maternal age patients. In this case, 60 patients over 35 years old were included in the study. Again, these 60 patients were randomized into 2 groups, one group where embryos were chosen for transfer based on morphology, and the other group where PGT-A was applied for embryo selection. And what we can see here is a significant decrease in the miscarriage rate in the PGT-A group. So PGT-A can not only improve pregnancy rate, but significantly decrease miscarriage rates. This study is very interesting because it's focused on the analysis on the advantages of PGT-A in reducing the time to pregnancy. In this study, more than 200 patients were included. These are advanced maternal age patients between 38 and 41 years of age. And again, randomized into 2 groups. The no PGT-A group, where embryos were chosen for transfer based on morphology, and the PGT-A group, only euploid embryos were transferred after genetic testing. And what the authors see in this case is a significantly higher live birth rate in the PGT-A group, almost double compared to the no PGT-A group. But what is more important is that not only more live births are achieved in the PGT-A group, but they are also achieved faster. The PGT-A group, the average time to pregnancy in the PGT-A group is 7.7 weeks compared to 14.9 weeks in the no PGT-A group. The last study I wanted to comment is this one by Tix and colleagues. This is a non-selection study. In this case, in this study, more than 400 embryos were included. And what was done in this study is that all these embryos were biopsied and analyzed, but the result of the test was not revealed until after the transfer. So those embryos were transferred blindly without knowing what was the result of the PGT-A. And then retrospectively, the analysis of the outcomes of the IVF cycle were compared between those euploid and aneuploid embryos. So those embryos that where the test revealed that had a normal number of chromosomes is this PGT-A euploid group, and those embryos with an abnormal number of chromosomes, once the result of the test was revealed, is this PGT-A aneuploid group. So what happened in the euploid group? Well, these euploid embryos, 82% of them implanted, 73% of them went on to have a heartbeat and a gestational sac, arriving to what we call clinical pregnancy, and 64.7% of them ended up with the live birth of a healthy baby. So what happened in the aneuploid group? In the aneuploid, these embryos that were identified as aneuploid, only 40% of them implanted, 23.5% of them arrived to the stage of clinical pregnancy, and none of them gave birth to a baby. So what this study is showing is that the value of PGT-A is not only to select the best embryo, but to deselect those embryos that will have no options to arrive to a live birth. To finalize, some take-home messages. By selecting healthy embryos with the right number of chromosomes to be transferred to the uterus, PGT-A can improve IVF success, reduce the risk of miscarriage, allow confident single embryo transfer, reduce the time to pregnancy, and avoid the live birth of a baby with genetic disorders. Well, thank you very much for your attention, and now I'm happy to take questions if you like. Thank you.
Hello. So we're just going to read out some of the questions. Obviously, for everybody else that's part of the session, then feel free to write something again on the questions and answers part. So one of the questions we've got here is, this lady looks like is 35 with secondary unexplained infertility. Would you recommend PGT-A testing after 3 failed IUIs?
Yes, I would definitely recommend PGT-A to all patients, because there is benefits in improving success rates and reducing miscarriage rates. So yes, for sure.
Okay. And typically, how long do the PGT-A results take to come through to patients?
In our lab, we have results ready in 10 days.
Okay. And then another question is, can somebody do PGT-A if they're using sperm or eggs from a donor?
Yes, sure. PGT-A can be performed in any kind of embryo, independent of the origin of the gametes who produced that embryo, and it is it could be interesting even in young donors, young egg donors, because the incidence of aneuploidy in egg donors is about 20%. So contrasting what you might think, even young women are producing eggs with alterations, and therefore the identification of the embryos with aneuploidies is very valuable, even in egg donation cycles.
Okay. This is quite a common question that we get from a lot of patients that are already at the clinic, and is it, can PGT-A testing damage the embryo?
Well, I would say that in expert hands, with an experienced embryologist that has been trained to do embryo biopsy, there is no risk for the embryo at all, especially if this biopsy is performed at the right stage of development. And this is after 5 days of embryo culture. After 5 days of embryo culture, the embryo is what we call a blastocyst, and this blastocyst contains hundreds of cells. So at this stage, the removal of between 5 and 10 cells does not affect embryo viability.
Okay. So this is another like scenario. One of our viewers has said, I'm 45 years old with 6 failed IVF cycles, and the most recent 2 were failed due to chromosomal abnormalities. How would PGT-A benefit me?
Well, if you say that you have 2 failed IVF due to chromosomal abnormalities, this is showing that probably the reason why you are not getting pregnant is because you are producing a high level of embryos with aneuploidies. So by testing all the embryos that you produce for aneuploidies, you will identify those that are free from these abnormalities, like that. Prioritize or identify or choose these normal embryos for transfer, and the chances of a euploid embryo implanting can be between 50 and 60% in, I'm not sure if you said, what was your age? Sorry.
45.
Yeah. So in this range, the options of getting pregnant per transfer will be over 50%. So it will definitely help you.
Okay. And can PGT-A detect in embryos all kinds of genetic disorders, or only a certain few? Or
no, PGT-A cannot detect all kinds of genetic disorders. Only those disorders caused by the presence of an extra chromosome or of one chromosome less can be identified in embryos through this testing. Other types of genetic disorders involving, for example, a point mutation or a change in one of the letters of the DNA is not going to be identified by PGT-A. There is other type of PGT that is called PGT-M, that stands for monogenic disorders, that would be required for identifying those type of genetic disorders in the embryo.
Okay. So I just want to give maybe a couple more minutes if anybody's got any more questions. So how many embryos? Okay, so in terms of like costings and things like that, because somebody's asked how many embryos includes in the free PGT-A. Any questions to do with that? What we can do is we can arrange like a follow-up email just to talk about like costings and what exactly is included and whatnot. So we can get back to you on that one if that's okay. Okay, so there's another question. That's nice. How can I overcome my chromosomal abnormalities?
Well, unfortunately, there is no way to improve chromosome abnormalities. So the chromosome abnormalities that an individual has is not able, we don't have technology to correct them if you want. So the only thing that we can do is identify which ones of the embryos that you produce have a correct number of chromosomes and choose them for transfer. In the case of having none of them, another cycle can be produced. And if that's the case, so that a person do not produce any healthy embryos, maybe it's something to consider moving on to using donor eggs or sperm. But yes, PGT-A is not a test that is going to fix the presence of chromosome abnormalities. It's going to simply identify if there are or not, and depending on the result, maybe the treatment can be managed so that other options can be offered to the couple in the case there is no healthy embryo to be transferred to the uterus.
Okay. Is it possible to biopsy embryos that are already frozen and not being biopsied before? Imagine the blastocyst is, or the embryo is in day 5 or 6. Yes,
sure. Those embryos can be thawed, biopsied, and frozen again. Now, most clinics, they have like a survival rate after vitrification or after freezing their embryos, which is very, very high. So even to freeze thaw processes are not going to affect the viability, or are going to affect very, very few of the embryos produced by a couple, because now the cryopreservation protocols are very, very good.
Okay. So we don't have any more questions at the moment. So this is your last little chance to squeeze a couple more in before we log off, I think. Also, don't forget, obviously, the final message about Mother's Month, that we've got this promotion going on with iGLS for the free PGT-A, which is valued at 3,000 euros. So it's quite an attractive promotion. So I don't think anyone else is going to ask any more questions. So I think we can close the session. I don't know, Maria, if you've got any final words, or is that it?
No, thank you very much for hosting this webinar, and I wish you the best in your IVF cycles. Thank you very much. Have a nice evening.
Thank you, everybody. Goodbye.
Bye.
Bye.